Welcome to Unfreeze Your Science, a podcast powered by CryoPDP, a temperature-controlled logistics company for the life sciences and healthcare industries. And now, it's time to unfreeze.
Hi everyone, thank you for having me for this opportunity and to talk a bit about what my laboratory does. So our main goal is really to transform, to repurpose immune cells as living drugs for a variety of diseases. This can be cancer, can be autoimmune disease, can be organ transplant rejection, can be allergy. And why such grand vision? Well, because it turns out that, the immune system is everywhere in the body, right? You have immune cells in your brain, you have immune cells in your limbs. Every organ has immune cells. Also, all immune cells, or almost all of them, can travel through the blood. And the blood is the most accessible tissue. And so we believe that if we can tap into this gigantic resource, we can purify immune cells, genetically modify them to give them new properties, and then put them back into the patient. So one big focus of ours has been type 1 diabetes. Type 1 diabetes is autoimmune disease. It's the body at war with itself, and more specifically, the immune system of this patient recognizes and destroys their pancreatic beta cells, which are responsible for producing insulin. Without insulin, you cannot control your blood sugar. And so these patients, they are dependent on insulin injection several times a day. They have to monitor their blood glucose closely. They can suffer blindness and even amputations. And so it's a major need. Also, I should mention that unlike type 2 diabetes, type 1 diabetes affects mostly children. That's when they start, that's when they get a diagnosis. And so, it's just very sad overall that these people have this genetic predisposition plus some environmental components. It's not exactly clear yet, what's the trigger in many of the cases. But so they have this condition that they have to live with for the rest of their life. There's currently no cure for type 1 diabetes. The closest that it's come to it is, other than the daily insulin injections, is a pancreas transplant. But then, of course, the disease started because the immune system recognizes and destroys the pancreatic cells. So even if you put a new pancreas in, it is also going to be destroyed unless the patient takes immunosuppressive drugs like tacrolimus and sirolimus. Now, these drugs work, and they've been used for decades now in the organ transplant field, so kidney, liver, lung. But the problem with them is that they're not specific. They shut down all immune responses, right? So now these patients, yes, they get to keep their, pancreas for a while, but then they become incapable almost to fight infections, the resurgence of blood cancers and other problems. And so the idea then would be to try to switch from immune suppression, just overall immune suppression, to something more targeted, something more specific. And so that would be the realm of immune tolerance. So immune tolerance is the idea that you can induce unresponsiveness of the immune system towards a specific protein, a specific target antigen. So for example, during pregnancy, right, mothers get pregnant with the fetus, there is 50% foreign to them, right? It's half mom, half dad for 9 months. Doesn't get rejected. Right? So somehow the human fetus has learned to induce immune tolerance in the mother's immune system. And so if only we could understand how this process works in nature, we could try to repurpose it to help treat autoimmune disease and also to solve the problem of organ transplant. So the way we go about this is we look at all the immune cells, and so there's one type of immune cell that really stands out. And that's a cell type called the regulatory T cell. So everyone probably has heard by now about T cells. T cells fight virus-infected cells. Could be COVID, could be the flu, could be anything else. T cells also fight cancer, right? There's now quite some therapies that are based on, supercharging T cells to find and destroy cancer cells. So these are most T cells. So T cells kill stuff. And then you also have helper T cells, which help the killer T cells kill unwanted cells. Now, of course, with such a powerful army, if you will, of the adaptive immune system, you'd want someone to keep it in check, kind of a general of this army. And so the general is the regulatory T cell, or Treg for short. So Tregs are T cells, like the name indicates, but they're dedicated not to kill, but to suppress, to inhibit specific immune responses. And so we know this is true from studies in mouse and human. So both mouse and human can have mutations in genes that lead them to not have these cells. And when it's true, they have widespread autoimmune disease. So that's how you know that, the brakes are off and the immune system does not know when to stop and starts recognizing and destroying multiple tissues. And so that's how we know that T cells might be important. Now, the importance is the first step. The second step is, well, okay, but can we use them? And the answer is what we're working on, right? So the field as a whole, so Tregs, they're not discovered that long ago. The first report in humans is 2001, so it's been like 22 years now, which is not that long in the field of medicine anyway. But so we know they exist. We can purify them. We have cell surface markers that we can purify these cells using magnetic sorters or fluorescence-assisted cell sorters, or FACS for short. So we can isolate them. And then we've also gotten very good at genetically modifying them. So this is, again, very good, but could still be better. But what we can do, nevertheless, is we can purify these cells, we can stimulate them, and we can modify their genome. And so why is this important? Well, this is important because it turns out that, Tregs are a quite rare subset of cells. If you have 1 million white blood cells from someone's blood, only 1% of those are going to be Tregs, right? So for every million white blood cells, you only get 10,000 Tregs. And so you can imagine if you're going to use these cells as living drugs to infuse them into a patient is going to become a challenge. Moreover, just like regular conventional T cells, Tregs have a wide array repertoire of specificities. So let's say you want to only expand the regulatory T cells that recognize antigens in the pancreas, right, to protect the pancreas from immune attack by this unwanted autoreactive T cells. Well, it's no longer 10,000 cells. Now you only have 500 cells per million or even less. And so this quickly becomes non-feasible, right? And so that's why we really took a page from the cancer research field, which is the field of CAR T cell therapy. So a CAR is a chimeric antigen receptor. It's chimeric because it's a mix, like it's like the Greek mythology, right? It's a mix of two different receptors. Part of it comes from B cells. It's an antibody-based part of the receptor that allows it to recognize any protein on the surface of a cell. And then you have a signaling domain. So signaling is just how cells talk, how cells make decisions. And so the signaling domain is inside the cell, is intracellular, and that's how the T cell is going to make decisions. And so taking these chimeric antigen receptors, these CARs, it is possible to redirect T cells and now Tregs to any surface protein we want. So the case of most success so far has been anti-CD19 CAR T cells for B cell leukemias and lymphomas, right? So B cells express this protein on the surface called CD19, and you can make a CAR T cell against that, so that every time this CAR T cell sees this CD19 positive leukemia cell, it kills it. And that's been really promising for leukemia patients. Now, we and others wondered, well, can we use this beautiful technology and adapt it to immune tolerance, right? Not to destroy something, but right to prevent something from getting destroyed. And so that's where the CAR Treg concept comes into play. Is the idea that you can take someone's polyclonal, so a bag of specificities of Tregs from their blood, and then you redirect them all towards a specific target with the CAR. So you could imagine like a CAR Treg that is specific to an antigen that is expressed in the pancreas. And so if that's the case, these cells can be tiny firefighters that go to the pancreas and stop the inflammation and destruction there. So to go about this, the first step was to get the Tregs, and that we can get Tregs, the regulatory T cells, we can grow them in vitro, right in the lab, we can expand them. We show that they work, so they suppress T cell responses and B cell responses and antigen presenting cells. So, Tregs, I should say, that's what's attractive about them as a living drug, is that, every small molecule binds to one or more molecules, but a Treg is a whole different level. The Treg, as a cell therapy, it can do 10 things at once, because it's what these cells evolved to do in our immune system, is to calm down different types of immune cells, to lead to tolerance when it's needed. So we take these cells and redirect them to a specific antigen. And so which antigen should we pick? So the first that we've been working on is called HLA-A2. So HLA stands for human leukocyte antigen, and you might have heard of this because there are large, databases of HLA alleles in bone marrow transplant databases. And why is that? Because HLA, turns out, is not just one gene. There are 3,200 flavors, alleles of this gene, and each one of us has 18 of them, 9 from mom, 9 from dad. And so what that means is that it's virtually almost impossible to find two people in the world who are matched. So this is why when someone is a bone marrow transplant or organ transplant, there's a gigantic effort into reaching out to, family, friends, relatives, because those are, the relatives have been the most likely to have more shared HLA alleles than someone across the world. But sometimes it happens, right? So all this to say that the HLA is an interesting gene to target because it's usually, the cause of rejection. It is the mismatch, right, between HLA alleles that is part of the problem, part of what leads to organ rejection between unrelated people. So in this case, we kind of, we turned the concept on its head and thought, well, can we, instead of using a mismatched HLA allele as bad news, can we make it into good news? And how do we do that? Well, we do that by targeting this HLA-A2. So HLA-A2 is an HLA allele present in 30% of people in the world, so it's pretty, ubiquitous. A third of people in the world have this allele, so then 2/3 don't. And so you can imagine a scenario where you get the pancreas, the islets, from someone who is HLA-A2 positive and transplant them into someone who's HLA-A2 negative. Or if we look a bit into the future and we're also working on this, you can take pluripotent stem cells. So embryonic stem cells can become any tissue in the body. And these are HLA-A2 positive, and you differentiate them in the laboratory into beta cells, into pancreatic beta cells, and then transplant those. So either way, the source of the beta cells, be it from someone or be stem cell derived, in the end, the idea is still you have beta cells that are HLA-A2 positive. And so once you have that, you then can have this as a hook, as a bait, that you can design CAR Tregs that recognize it and specifically traffic there. And so after, a couple years of trying this, we're able to design and generate a CAR, a chimeric antigen receptor, right, that recognizes HLA-A2, and we're able to integrate it in the genome of Tregs, of human Tregs. And then we show that these Tregs are suppressive in the laboratory in vitro and also in animal models. So we have what's called a humanized mouse. So these are immunodeficient mice. They don't have their own T cells or B cells, so they don't reject human cells and tissues. And so you can take these mice and you can transplant to these mice human pancreatic islets, and they make human insulin. And luckily for us, human insulin works on mouse tissues. And then we can then put in human immune cells that will reject the islets, right? And then human CAR Tregs, which should prevent the rejection of those islets. And so what we've shown is that you can take these humanized mice, transplant them with human HLA-A2 pancreatic islets, and we use in the kidney capsule because the kidney is a highly vascularized site, and it's easy to expose and then transplant there. And then we intravenously inject, so just like we do in humans, intravenous injection of immune cells and of CAR Tregs. And we show that the CAR Tregs, they suppress immune responses in the mouse, so the mouse don't get sick from immune rejection, and they traffic to the islets. And so this is very important because, again, one I mentioned earlier that one disadvantage of small molecules like aspirin, just to give an example, versus a cell, is that the small molecule can only bind to one or two molecules, right? It has a limited, range of actions. That's number one. Number two is that the small molecule, if you take it orally, for example, it just goes everywhere in your body, right? So the cells are different. Even if you inject the cells, intravenously in your vein, they can traffic. They have a zip code system, if you want. They traffic and they go only to the place where they're needed, right? Because these cells, the immune cells and Tregs in particular, T cells in general, they need stimulation to stay alive. They need something to get busy with. So if these cells are not being stimulated anywhere else in the body, they don't even go there. So that's kind of what was beautiful to see in the animal model, is that over time you see an accumulation of the cells in the islets, in this site of action. And so that will make for a both safer and more efficacious therapy than if you're taking something like taking the exam, again, the case of cancer, like chemotherapy or radiotherapy. Radiotherapy is a bit better now, right? You can focus the beam, but chemotherapy, your whole body. That's why the patient loses hair, their blood cell counts go down, because anything that divides fast, the cancer chemotherapy is going to attack, right? So it's a non-specific blanket therapy. And in the case of diabetes, type 1, transplant rejection is these immunosuppressive drugs that, once again, they shut everything down. Now, the promise, the big promise of Tregs is that you can now make it specific. You can shut down this specific unwanted immune response in this specific place in your body. And so we've shown so far we're able to generate this chimeric antigen receptor construct. We're able to genetically introduce it in the genome of Tregs. And what we did is actually we used CRISPR-Cas9, which is a way to modify the genome of cells and also to be precise. One of the dangers of early gene therapy is that people made genes to go into human cells, but didn't get to pick where they went. They could go anywhere in the genome. And so you could imagine that if it goes in a part of the genome that is involved in, stopping cancer's growth, it's not great news if your gene goes there, right? It's going to interrupt an important process. So with our technique, we do precision engineering, right? We're actually able to introduce this chimeric antigen receptor gene that recognizes HLA-A2 specifically in the gene locus in Tregs. That's the T cell receptor locus. And so that's great because this is where the gene for recognition should go. So all the checks and balances that T cells have in a genomic location can now be transferred, be applied to our synthetic gene. So we have these cells, they're purified from human blood, they're genetically modified to have this CAR against HLA-A2 in their genome in this precise location of the TCR, T cell receptor locus, and then we tested them in vitro and in vivo. Now, of course, a couple next steps, right? That's when our work is not done yet. So one thing we're doing is we're further improving on the design of the constructs. It's actually part of the fun of synthetic biology, synthetic immunology, is that, you get to play with these cars and we can always try to make them better. Can change the amounts of protein you have. You can change how strong or how weak the signal is. You can change the target. HLA-A2 was a good starting point, but as I mentioned, not everyone has HLA-A2. So you could imagine trying to go after another target that maybe all pancreases have, but other cells don't, right? And this way you could make more universal treatment, if you will. And speaking of universal too, that's kind of a third step is, right now this will be an autologous therapy, so self. So you take your own Tregs, modify them, put them back in. But you could imagine this is not very scalable, right? These patients are already suffering. Maybe their Tregs have some defect to start with, right? Because if they have type 1 diabetes, they have an autoimmune disease. Maybe their Tregs are not, the most potent cells there could be. So what if we could instead take regulatory T cells, Tregs, from someone with a non-diabetic control, and then can we make them universal? Can we change genes in them that make them cross the allogeneic barrier, right? So you can use the same cell for multiple people. So it's kind of a universal cell therapy idea where you make hypoimmunogenic cells that are kind of invisible to the immune system, so they don't get rejected, and that will allow you to expand on this idea, right? You can massify it, you can make it more accessible by having centralized banks of cells they can use. And this doesn't have to be, I work with the regulatory T cells, but this could be cardiomyocytes for the heart, it could be adipocytes, could be anything, right? I think it's another powerful idea that we have a little bit of work on to make universal cells for cell therapy that could be applicable across multiple people. And so this is what I've been doing, and then I like also to mention that this type of work is only possible because there's great logistics out there. You can imagine work with human tissues and human cells, right? We get, I'm currently located in Charleston, South Carolina, and our blood comes from Memphis, Tennessee, or from Washington State, or from wherever, and our islets also come from California, from Florida, from UPenn, Pennsylvania, right? And so there's quite some effort, because when we work with lab animals, you get access to any tissue you want, right? The animals are in our laboratory. we can extract any tissue you want. But when you work with human tissues, right, it becomes more complicated. And so it's really been, a great privilege as well to be able to, work with human islets and human blood cells and put them together in a laboratory and see how they interact. This is a multi-consortium. So for islets in the United States, there's the IIDP, Integrated Islets Distribution Program. For blood, there's multiple companies as well, mostly commercial. But so the idea again is that, work with human cells requires a level of coordination of receiving fresh tissues that we can use for functional experiments. Because that's the thing I would say is that some human work has to be more descriptive. Maybe post-mortem, you look at the tissues, patient samples, but what we do is functional, right? We get human blood cells killing or protecting human pancreatic cells that make insulin and prevent the blood glucose from going too high in these animal models. And so to see human cells, how they function, do the functional assays again, it's quite it's quite a privilege, I feel like anyway. It's quite a privilege to be able to do that with human cells. Yeah. Thank you.
Thank you for listening. We hope you've enjoyed this episode. Stay tuned and discover more about the work of our guest in the description. Podcast powered by CryoPDP, a temperature-controlled logistics company for the life sciences and healthcare industries.